So our next talk is from Dr. Elisa Oltra from the Catholic University of Valencia. And Elisa is going to talk to us about differential microRNA profiles in PBMCs and plasma EVs, which are extracellular vesicles of severely affected ME patients.
Good morning, everybody. I want to present you this morning the work that we've been doing during this past year to try to compare the profile of microRNAs. Where's the pointer? It's in the middle. Okay. The profiles of microRNAs in mononuclear cells in the blood. With those same profiles on the extracellular vesicles present in plasma of severely affected ME/CFS patients. This work has been possible thanks to the funding of the Rnas Research Fund through the ME Association. I have to say before starting that this proposal was initially based on a previous work by our group that was published back in 2015 in the journal PLOS One. We were the first group to perform microRNA profiling, genome-wide profiling in PBMCs of fibromyalgia patients affected of chronic fatigue. So this time we wanted to take a step further and not only check those profiles in PBMCs, but also in another fraction that could give us more information because these extracellular vesicles, as we will see in a few moments, they come from all the tissues in our body. So before we start, I would like to briefly introduce microRNAs, although yesterday we already were introduced these small molecules, and also extracellular vesicles. And no. So it's this one. Okay. Nope. No. Sorry. Yes. Okay, it's reversed. Okay, all right. So, briefly, what I am showing here is that these small and powerful molecules are epigenetic regulators of the sequences in the genome on the sense that they can bind to the templates that produce proteins. You all know that proteins are the effectors in the cell, and that depending on the proteins that the cell makes, it will have specific status and a specific performance. So these are molecules that generally are in all our cells in our body at different ratios, and they are produced in response also to different hormonal changes in different responses of the cell. So because of their property, they have been shown to be good candidates as biomarkers for different complex diseases, and that was the reason we initially considered them as a potential fraction to be evaluated. There is a complex machinery, a complex process involving several proteins that give a place to these molecules. So our interest is to know what is the presence of these different microRNAs. More than 2,000 have been described in humans now. And in reference to extracellular vesicles, here I'm showing a cartoon of a cell in which we can see that there are different types of vesicles that the cells in general produce. They produce vesicles that just correspond to waste, and others that have specific features that have been tagged as exosomes because of certain markers, and that are considered real carriers of communication for long distance spreading of disease, and also for communicating at long ranges within the body. So these different type of vesicles, and especially the exosomes, can give a hint of what is the status of the cell, And they package different type of molecules. Among the molecules that they are carrying, you can imagine that are the microRNAs of our interest. So these vesicles are released into the bloodstream not only by the cells in our body from different places, brain, gut, any other organ that you can think of, but also by the microbiota in our gut. So it will be possible, in theory, to get this fraction and evaluating what is the content, have a liquid biopsy of this organism. So it will be, that was the reason that made us think that it was very interesting to compare these two fractions, the content in microRNAs in these two fractions. So to do this and to do it the best way possible, we selected samples from the UK ME-CFS Biobank, which is very stringent in the diagnosis of patients that have to comply with the Canadian consensus criteria, and this way of diagnosis also guarantee that we minimize patient heterogeneity in the group that we were going to study. Not only this is important to minimize potential bias, but also, for the potential bias, due to analytical variables, taking samples from a unique source, and the way in biobank is usually operated is using these standard operating procedures that are highly optimized at all levels, at the level of collecting, at the level of processing, and also storing the samples. So this would grant the optimal quality, which was something that we could test afterwards. So what was the demographic description of the cohort? I have plotted here most of the features. You don't need to read all these numbers. Just I want to mention that the group was 15 patients and 15 healthy controls. This is a small group, as we have been said in the previous talk. It's not desirable to go to small groups, but I have to say that in this type of analysis, the funding resources are the limitation. So when you are also in an initial phase of discovery or of evaluation research, you need to also try to adapt the resources to this initial phase by starting with small numbers. If these pilot studies turn out encouraging, as I think I will show you this is the case, then we should be ready to step up with a larger number of participants to give this power that we were told a few minutes ago. Okay, so what mainly this describes as well is that the cohort study had a significant difference between the patients and the controls, particularly on the physical side and not on the mental side. And this is another questionnaires that are also recorded at the biobank for completeness of the data that is associated with the sample. So this is very important, as we were told yesterday, that you can then try to correlate the variables, the molecular variables that you are studying with these clinical parameters or with these other parameters. Okay, so regarding the clinical parameters, we have that from the data provided from the biobank of the 34 parameters, 30 were complete for the 30 individuals. And when evaluating them, we found something quite interesting because we had been reading, not a physician, but I've been reading about repeatedly about the paucity of clinical markers for CFSME. And then we were surprised that just by relaxing, this is true, by relaxing a little bit, by relaxing a little bit the restriction of the statistical test, we could get four different parameters that were significantly different between the two groups. And in the case of the creatine phosphokinase, this was really different, so different values between the ME/CFS group and the healthy controls. I mean, this is quite interesting and worth, I guess, to pursue, but this is only this is only from our small cohort, so we don't know if by enlarging these numbers, this may stay or could vary as well. I just have to say that this enzyme is linked generally in the clinical practice to muscle damage, but it is a resource to store phosphate, so it could be interesting to follow this up later. So now we're at the step of how do we isolate these extracellular vesicles is going to make also a difference on what the content is. Let's say that if we all use the same procedure across different labs, we could compare these results. But what happens in the methods available is that there are many different of them established with different outcomes, of course. The most, the method that gives the most pure fraction is applying differential centrifugation and also followed by density gradient centrifugation. But this technique, as you can see here by the number of hours that are required to get the vesicles purified, is not practical to be translated into the clinic. So there are also other that are high cost associated because they use antibodies, and then there is this other which are based on size exclusion or filtration and precipitation. Then we try to decide between these two firstly, but then what we found is that this group that is cited here had already done the comparison of all these different type of methods and convinced us to see that the precipitation method was allowing recovery of 90% of the vesicles present in the plasma. So, and also consistently. So the strategy to check on these extracellular vesicles could be two different. Either you get a very pure fraction and just concentrate on that, which we didn't take, and then this other one, which is we precipitate most of the vesicles, perhaps some of the vesicles not relevant, but we have the big picture, not to miss anything. And also in a method that could be translated to the clinic. So if it reports something, it's ready to be implemented. So this is what we decided. And here you have, after we purify the vesicles, you have an image of how the profile of these vesicles look like in either of the groups. And some of these variations are not very significant, but when we checked in the whole group of samples, what are the number of particles that the same volume of plasma yields, we find differences in the way that the ME/CFS group present with larger number of particles. This was recently, a few months ago, also published in this sense, an increase of the numbers in ME/CFS group in another cohort studied in Barcelona in Spain. So it goes in the same, it is nice that it fits with previous data. And we went a little bit further, not only checking the number of vesicles, which, by the way, I have to mention that an increase in number of vesicles has been in general associated with disease. Okay, and we also check on the size. And the average size is also different, significantly different between the two groups, having a smaller size in general in the ME/CFS group. So, because the method that I told you about, precipitating the extracellular vesicles, can be performed in the presence of an enzyme that is called proteinase K, or in the absence of it, we also perform the isolation with this other alternative technique. The reason behind this was that in the plasma, as I told you before, these vesicles can be, there are also aggregates that could be confused with vesicles. So to get rid of these possible aggregates that are not vesicular in nature, and also to get rid of some microRNAs that could be fixed to the vesicles on the outside of the vesicle through protein interactions, we decided to also check our fractions for all downstream applications with proteinase K. So you will see in some of the figures minus or plus proteinase K. And the point here is that by treating with this enzyme, we still found the same trends. The trend is that the group of ME/CFS patients have larger number of particles and smaller size. So because of this, we went to look to another parameter of these vesicles. The parameter consists in measuring through an instrument, particular instrument, the electronegativity of and the electric potential of the membrane of these vesicles. It has been in general shown as a reference that the electric potential of plasma membrane in general is around -8 millivolts, and in the case of exosomes is increased in negativity to -15 as an average. These reference values are set in the context of giving that negative property to the communication properties of these vesicles as well. And what we find here is that in general, these vesicles that we are purifying from the plasma of patients, they are slightly more electronegative, either indicating that are richer in exosomes, which increase this electronegativity, or there is something else associated with them. Here there are some controls in which when we treat with proteinase K, it seems that some of the electronegativity in both groups, but this is just a control, so not. So, I prepared some videos so that you can see how these vesicles look like. And I want to take the attention that these vesicles have been prepared by exactly the same method from exactly the same starting value. Also tell you that in addition to all the parameters that I told you about, the technician that runs this instrument, she was, you see that is a person that is running 8 hours a day samples from different groups, so she really has the eye for this. She told us that she thinks the vesicles have some refraction properties that are not the same, but this is not quantitative. We quantitated as much as we could. So this is, so how do I play the video? Is it, they play here? And here at the same time? Okay. So this is what this nanosite instrument records. And by doing this, it records sizes and numbers. So, okay, so you can clearly see that the image is quite different. We also went and took a look of the vesicles we had prepared, both at the level of protein expression related to markers of the exosome subgroup of vesicles, and also to look for this characteristic of double membrane that we can see by electronic microscopy of these samples. And then we were ready to prepare RNA to do the real experiment that we were proposing to do, that is the comparison of this microRNA profiling. When we check on the RNA yields and quality control, the quality that assesses how good the RNA is obtained from this fraction of PBMCs is called RNA integrity number. It's a number that ranges from 0 to 9, and this number reflects the higher the value, the better the quality it is. So from the 30 samples we received from the UK Biobank, we found that 28 of them were high quality. There is always some samples for some reasons that do not perform optimally, but this could also be due to the procedure itself. So, and also on the EVs, you don't have this parameter because the vesicles do not contain that much ribosomal RNA that is the reference molecule to determine this this quality control parameter. We also look at the profiles corresponding to the values that I told you at the long range, and also enlarging the size of the small RNA that was of our interest from 20 to 60 nanometers. And in PBMCs, you can see that you have a peak because the content is high and the variability is less. In EVs, on the contrary, we have a very high complex mixture of RNAs that is contained within these vesicles, but this has been reported before. So we were on the right track regarding quality control of our samples before we proceeded to study them. Now, how do we do the profiling? There are also different methods, and in fact, we think that the optimal method, if you have the possibility of bioinformatician to interpret the data, is RNA-seq, because then you completely sequence the whole RNA there. But we, in the case of our project, we have also a restriction in the fund the amount of money that we had available, and these type of assays are quite expensive. I can tell you that a chip of this type, it holds 12 samples and is over 4,500 euros. So it's quite expensive. And the technology is very, it's the latest technology to measure the amount and the variability of microRNAs in not on a solid support, but is on suspension, and to measure it directly. So for microarray that you have been used to probably to get the data from this type of profiling studies, in the microarrays you have a solid support where you have the probes fixed that hybridize to the complementary of your RNA. So your RNA has to go through some enzymatic reactions which introduce bias. This company, what has developed is a fluorescent tag that is complex on the way that it allows to hybridize to the target without amplification. So the fluorescent target hybridizes directly to the microRNA, and what you're counting here is the number of molecules that really you have there without any amplification. And for that reason, this type of data does not need validation by an alternative data. So we thought this was a very interesting approach, and in addition to that, the instrument, which is cost over $250,000, it was available through one of our collaborators. So taking advantage of that, we performed the profiling in both fractions, and what we see here is after normalization and after restriction analysis, so that we can get rid of those microRNAs that are not showing in some of the samples. So we were very stringent because those microRNAs with less than 75% of the samples fulfilling the threshold set here after normalization, which was quite stringent, were discarded. So from 800, starting from 800, we discarded over 600 of them. We found that 42 were exclusive of PBMCs, and we found that 101 were exclusive of EVs, with a more complex mixture of microRNAs in EVs than in PBMCs. And as a whole, there were 49 that were shared between the two. So these 49 could refer to microRNAs that are in vesicles because are derived from the PBMCs, right? Okay, so when we filter to have statistically significant differences, we even went down with these numbers. So we, of the differentially expressed, microRNAs in PBMCs, we finally, what has happened here, sorry. I would, okay, here. So we had 11 in microRNAs overexpressed and 10 in microRNAs underexpressed. And in the case of EVs, we had these two sets, which also were narrowed down to very few microRNAs being expressed differently. Okay. So in the table, I have represented that after doing these stringent analysis, we even went further and removed outliers according to Chauvenet criteria, in which an outlier was considered those values that surpassed the 2 times the standard deviation of the mean value of the group. So there were some of them that were also set outside of the selected ones. And then here I can show you that the difference between groups show statistical significance. As I was telling you, only 7 of them fulfill this criteria into the microRNAs isolated from PBMCs. And then for the upregulated, there were a few more, okay? You don't need to know these numbers, but some of them also coincided with the ones that were found by other researchers, as Alain said yesterday, the 150 and the 140. Okay, and we, even being that the nanostring was not is a technique that doesn't require validation, we still went and selected a few of them and validated by another alternative technique, which you can see here. Okay, the question now is, with this data, can we separate both groups? And this is another type of analysis that is called principal, sorry, I don't get this, principal component analysis, that you can see that, I mean, not completely well, but with a combination of some of the microRNAs, we could certainly differentiate to a certain point both groups. So this is exactly the same type of analysis that I was showing for the PBMCs, but now with the EVs. And in the EV group, in the extracellular vesicle group, we found that it was predominant, the group that had the microRNAs upregulated, even to 2.6-fold the values of the healthy controls. And again, I wanted to show you how these individual data look like, you know, the distribution. I think it's important to see if they are considered significant, statistically significant or not. And both groups, again, the former was in the underexpressed, this was the overexpressed, which took 2 slides because they were much larger in number. The separation, according to this principal component analysis, is not as good with the combinations we have made so far from the extracellular vesicles in reference to the PBMCs. So this would argue for the PBMC profile to be better option, to be more specific for differentiating both groups. But we have many more players in the EV set. So perhaps the EV, the extracellular vesicle fraction, is more sensitive to changes. Okay, so once we get all this list of microRNAs, one wonders, what do these mean? What is the significance of these results? And we used one type of okay, sorry. Again. Okay, we used this type of software called Diana Tools, which use a database to kind of relate the microRNAs that have been found with possible gene targets. Only in the case of tarbase are targets that have been experimentally validated, so to be really stringent again. And it caught our attention that in all the 4 groups, PBMCs underexpressed, PBMCs overexpressed, we find this neurotrophin TRK receptor as one of the pathways that is chosen by this software. And also this pathway appears, we first thought it was a mistake because we analyzed this data just a few days before I came here. So we are at the point of checking the list of the genes included in this pathway in each one of the 4 options, because obviously the microRNAs introduced are not the same, and thus the genes may vary, at least some of them. So in conclusions, we wanted to know, okay, if we have these microRNAs in extracellular vesicles that come from any type of tissue, could we know by the presence of the particular microRNAs in this fraction that are overexpressed, if it's some tissue in our body that is releasing this microRNA? So reference values are very important, and databases are very important. This has been repeatedly said in this meeting. But this in particular was non-existent until 2 years ago, and is an atlas, a tissue atlas of what is the abundance of microRNAs in different tissues in the human body under normal conditions. We have this type of tissue atlas from many resources and built up for long as a database, but for microRNAs it was not available until 2 years ago. So we checked this, and what we find is that the main tissues that appear to be more rich in the targets that we have obtained in the profiles, the microRNAs that are differentially regulated, come from thyroid, from fascia, muscle, and also brain and vein. So I think this is a good resource to follow on the genes, and because some of the microRNAs also coincided with a previous report of these authors leading microRNA and the machinery of producing microRNAs to post-traumatic stress disorder, in which they show this machinery is downregulated. We took a look, but not on the samples of the biobank, because that was not programmed, and then we cannot use it unless we ask for permission for extra addition of these markers. But we checked in a local cohort if this could be the case, and surprisingly, what we found is that for all these members of the machinery, we got upregulation in any case, not downregulation, so it's different. So we also have been working on DNA's methylation pattern, and the last thing I want to say for future directions is that we need to put together all this information that is coming up from different small projects that have been designed in a certain way to be available to, to be available to build up these databases that we heard a few minutes ago. So we have some local bioinformaticians. This is the company that has interest also in checking and what this microRNA combination could mean in the sense of modeling the disease. And we want also to go in the way of, to understand much better what these microRNAs that we found out of the regular levels, what do they mean? So we need to take other steps to find out what do they mean, and also to build up in numbers. And this is the group in Spain and the collaborator that I told you, Dr. Nathanson in the US. And I want to thank you all
for your patience.